We had 3 posters that were presented at SITC. 2 of them were about the combination study where we're using MRx0518 in combination with Keytruda in patients who have developed secondary resistance to Keytruda. So that was 2 of the posters. And the 3rd poster was about a new study where we're using 518 as a monotherapy in a very different patient group. So I'll give you an update about the combination study first of all. So we've completed part A of that study, which is in 12 patients with advanced metastatic solid tumours, renal cell carcinoma, non-small cell lung, and bladder cancer. And these patients, if you remember, are actually patients who have developed resistance to prior checkpoint therapy, so therapy like Keytruda. And in that instance, they'd be very unlikely to respond if re-challenged with a checkpoint therapy again. So what we are doing in that study is to look at the combination of 518, which is an immunostimulatory live biotherapeutic that we've developed, with Keytruda in these patients to see whether we could generate an anti-cancer response. So these patients have all got progressive disease when they come onto the study. They've all run out of approved treatment options. And what we saw in part A was pretty remarkable, really, given that we wouldn't expect them to respond just to Keytruda alone. We actually saw a clinical benefit in 5 out of the 12 patients on that study. And those patients who did respond or had long-term stable disease actually had a very durable response. So we've had patients on study now for more than 12 months on that combination, and the safety profile looks really good with the combination. We have no serious adverse events related to treatment. So that update was really important because it shows and confirms that proof of concepts that we talked about earlier in the year. And so the second poster on that study was really about what we're doing next with the combination treatment. So we're now into part B of the study, which is much larger. It's actually going to be up to 120 patients. We've expanded the clinical sites. We started the study at MD Anderson. We now have 4 more sites in the US. And we've also expanded the tumour types. So we've got kidney cancer, we've got bladder cancer, we've got non-small cell lung cancer. We've also introduced head and neck, triple negative breast, and MSI high tumours, all of which are in patients who have developed this secondary resistance to checkpoint therapy. So we're recruiting well, and we're expecting to have fully recruited that study by the end of next year, around about October, November time. So that's really important. We're really pleased with the progress that we've made there. And obviously, this is looking at 518 in combination with another drug. So one of the questions that we're very interested in understanding is what sort of activity will 518 have on its own? And whilst the combination study is being conducted in really patients who have run out of treatment options, we have another study underway, which is a neoadjuvant biomarker study of patients at the other end of the spectrum. So these are patients with solid tumours who have just been diagnosed with that solid tumour and will be scheduled for resection, so surgery to remove the tumour. What that gives us is really a setting which is quite unique. So these patients haven't had any previous treatments. They have a biopsy to actually diagnose the cancer. So that gives us a sample of the tumour that we can look at. And then they're scheduled for surgery a number of weeks later. And that gives us this window to actually treat with 518 before the surgery, when we can get additional tumour samples when the tumour is removed. And that gives us a really powerful way of looking at what 518 is doing in the tumour following that treatment period of 2 to 4 weeks. And we reported on this data at SITC, and what we're seeing, we think, is quite remarkable. So with a single strain live biotherapeutic that's being dosed orally, don't forget, we're seeing changes in the tumour even after 2 to 4 weeks treatment. So for instance, we're seeing an increase in immune cells infiltrating that tumour, cells which you'd expect to be associated with an anti-cancer response. We're also seeing changes in the tumour which are actually in the tumour cells themselves. So you look at the gene expression pathways in the tumour environment. We're seeing a difference there. And the gene expression pathways that are being upregulated and influenced are the ones that you associate with an anti-cancer response. And we're also able to detect changes in blood samples from the patients following treatment. So we're seeing an elevation of a couple of cytokines which are associated with an anti-tumor response. And this is really important because it gives us a lot of information about what 518 is doing as a monotherapy in patients. It's also incredibly consistent with what we saw preclinically, which is important because it demonstrates that our platform that allowed us to discover 518 in the first place is giving us a really good steer as to what to expect in the clinic. And it also allows us to have very constructive discussions with regulators so we can understand the contribution that 518 could make when it's used in combination with other therapies. And obviously it also adds to our knowledge about the product, which helps us when we're talking to potential partners as well. The other thing that we've seen in this study, and I should just say this is in the initial group of 17 patients that came onto the study. The other thing we've seen here, as per the combination study, is a very, very good safety profile, which augurs well for the future of 518 in terms of being able to be used long-term and with other therapies. So, we've got a lot going on at the moment with the combination study and with this monotherapy study, but in terms of where we go next, we're looking at ways of potentially evaluating MRx0518 with other types of checkpoint therapies in the clinic. We're also looking, we'll be looking preclinically at different combinations of 518 and other drugs. And we're looking at different treatment settings as well. So obviously, when you start off developing a cancer product, it's always focused in what we might term last line patients who've run out of treatment options. That's just where you start. But as you start to build more evidence of efficacy, we can look at different settings, potentially like maintenance therapies and other tumor types in earlier treatment lines. So I hope that gives you a good feeling for where we're going with 518.